Hello students, today we are going to discuss on transgenic animals and what are all the research ethics that are associated with the development of the transgenic animals. This is an outline about introduction to transgenic animals, methods of creation, some of the examples of transgenic animals, ethical objections and religious views on development of transgenic animals and conclusion. Transgenic animals. The animals, those have DNA from another source, is inserted into their genome are called GMO or transgenic animal or genetically modified organisms. Many transgenic animals have been created so far by biotechnology. Currently, no transgenic animal or product is approved by the FDA for human consumption. Why to create the transgenic animals? Some of the goals of the transgenic animals creations are it is to improve livestock of the animals, use of animals as bioreactors, production of pharmaceutical drugs used as a model organism in the research. And this is the motto for the development of transgenic animals. Creation principle. So how to create an transgenic animal. Inserting a foreign gene into an animal is called transgenic creation. The inserted genes are called transgenes. The genes must be transmitted through the germ line from one generation to the next generation of the particular animal. So every cell will contain the same modified genetic material in its genome structure. That is, the modified gene or inserted gene will pass to the next generation after the development or insertion of the foreign gene into the parent. So especially it occurs in the ovum or the germline of the animal. There are 4 basic methods in order to insert the foreign gene into that particular animal. So which involves the DNA microinjection. Directly the DNA is injected into the ovum region, and retrovirus mediated gene transfer, and next one is embryonic stem cell mediated gene transfer and linker based sperm mediated gene transfer. The success rate is very low in certain cases of sperm mediated gene transfer. DNA microinjection. The desired gene is transferred to the pronucleus of the reproductive cell. So here we can see the micropipette that holds the ovum tightly. This is a holding pipette on the left side, and we can see the micropipette is piercing inside the ovum, which is a DNA containing solution. It is a fertilized mouse egg to which only our gene of interest is injected with the help of our micropipette. Hence it is termed as microinjection. The manipulated cell is cultured in vitro in the embryonic phase, then it is transferred to the uterus of the recipient animal. The next method is retrovirus mediated gene transfer. Retrovirus mediated gene transfer is that the retrovirus is taken, the genetic material is inserted into the host cell to produce the chimeras. Chimera means copy of the cells. The chimeras are then inbred until the homozygous transgenic offsprings are born in this case. The next one is embryonic stem cell mediated gene transfer. Isolation of totipotent stem cells from the embryos. Then desired gene is inserted into these cells. The recombinant cells are then incorporated into the host embryo. Here we can see from the diagram the DNA is taken up and it is inserted to the embryonic stem cells. It is selected for expressing the desired gene. Then the transformed embryonic stem cells are injected into the blastocysts of the inner cell mass of the uterus. The implant is then placed in the uterus. Another method is that the desired gene is taken up. Desired gene is inserted with the vector to the fertilized egg. The pronucleus is created. It is implanted into the uterus along with the stem cells, embryonic stem cells. The foster mother or the surrogate mother will carry the implant and test offsprings for the presence of the desired gene. The male heterozygous offspring produces homozygous transgenic strain. The next is our linker-based sperm-mediated gene transfer. The sperm carrying the foreign gene, it fertilizes the egg where the foreign gene is incorporated into the genome directly. These are some of the examples of transgenic animals. So dairy transgenic cattle, the dairy cow carrying an extra copy or 2 types of casein gene produces 13% more milk protein. This milk is more nutritious. So currently the milk from these animals are under FDA review. That is Food and Drug Regulatory Authority of USA is still under reviewing this for the approval. The next is EnviroPig. EnviroPig is a genetically modified pig. It can digest the plant phosphorus more efficiently. It has the enzyme phytase in the salivary glands. The phytic acid in the pig meal is degraded to release the phosphorus, which is absorbed by the pig. Pig waste is the major pollutant. Next is transgenic fish. Salmon or trout can grow up to 6 times faster than the wild type fish because they have extra copies of growth hormone gene. The ornamental glowfish have red, green, yellow and orange fluorescent color. They are available as pet. The next is our transgenic mice. Transgenic mice are widely used as a model to detect gene expression. It is used in knockout technology to detect the gene function. The particular gene is mutated so that what is the role of the gene that can be identified by using the animal model. We call it as knockout mouse. Transgenic monkeys. Andy is the first transgenic rhesus monkey born on October 2nd, 2000 and has a green fluorescent protein gene. This gene incorporated into the monkeys. The monkey is capable of glowing under ultraviolet light due to the presence of this GFP protein, that is green fluorescent protein. Next one is transgenic rabbit. The transgenic rabbit is called Alba, named. It is having EGF, that is enhanced green fluorescence protein. This green fluorescence protein is incorporated into the rabbit by transgenetic mechanism. The bunny, which is created in 2000 as a transgenic artwork, it has a GFP protein that glows when exposed to the blue light. Previous one is ultraviolet light. This is an enhanced GFP that glows in the blue light. Transgenic cats. The cloned transgenic cat contain red and green fluorescent proteins which glows in the presence of ultraviolet radiation. The next is transgenic goat, capable of producing spider silk. Spider gene is taken and the silk gene from the spider is transferred to the goat. Each goat produces several grams of silk protein in her milk. But we have an alternate. Need not to modify because the goat are utilized for human consumption too. This gene can be incorporated into a bacterium for the harvest of silk gene, or it can be incorporated into silkworm, whereby it can produce larger quantity of the spider silk. There are some ethical concerns on the usage of the animals. The use of animals in science is a controversial issue. We have discussed earlier also. There are some moral and ethical factors contributing to the genetic manipulation. Even the biotechnological research seriously reviews on the ethical aspect of the animals usage in the research purpose. The Animal Welfare Concerns, the Royal Society for the Prevention of Cruelty Animal, that is UK RSPCA, in short, looking out for the animals since 1824. The use of animals in research causes greater suffering to the animals. Using animals for the production of pharmaceutical proteins, we reduce them to mere factories. So the animal should have the same basic right as the human being. Each biological species has a right to exist as a separate, identifiable entity in this world. Moral status of animals. Pets are the part of family. They should be given respect. They have a sense of pain and pleasure. The moral codes of ethics should be followed while using the animal in research. What is the boundary between natural and the unnatural? There is a boundary is constructed between what is considered as natural and what is unnatural. Crossing of species boundaries is unnatural. This becomes problematic when higher life forms are involved, that is, human-animal chimeras. Consequences of genetic modification. The consequences of genetic modifications might be positive or negative. Risks are there for human health and environment. Environmental safety issues. Modified animals might escape and breed with other animals. So transferring the new genes to the other population also happens. The retrovirus might infect the other organisms. Wider effects of producing disease-resistant animals will develop. Crossing the species boundaries. Crossing species boundary is a violation of the God's law. This scenario is changing the species concept regarding ecology, morphology, phylogeny, and evolution. Xenotransplantation issue. Why the xenotransplantations are carried out means the transplantation of the organ from the animals to human can cause zoonotic disease such as mad cow disease. The introduction of these diseases to human population could have devastating consequences. Ethical decision making. Some uses are generally acceptable, often because of their cost, usually welfare, health, and environmental, are perceived as negligible. Some uses are acceptable only whether the benefits are sufficiently great and outweigh any perceived cost. The Banner Report. In 1995, the UK Ministry of Agriculture, Fisheries and Food delivered Banner Report. It has 3 principles. Harm of a certain degree ought under no circumstances to be inflicted on an animal. Any harm to an animal must be outweighed by good, which is realistically sought. Any harm should be minimized as far as possible. These are some of the guidelines, CPCSA guidelines provided, that is common for committee for the purpose of